The uterine cervix plays a critical role in a successful pregnancy by acting as a strong, protective barrier that keeps the baby safely in the womb until it is time for birth. For years, scientists have understood that problems with the cervix can lead to devastating outcomes, including miscarriage, cervical insufficiency, where the cervix opens too early, and preterm birth. However, the precise biological reasons for these failures have not been fully understood. A recent expert review published in the American Journal of Obstetrics and Gynecology sheds new light on this topic by exploring insights from a cutting-edge technology called single-cell analysis. At its core, this research moves beyond looking at the cervix as a single piece of tissue and instead zooms in to study the function of each individual cell. This detailed approach has led to a major breakthrough: the discovery that cervical cells are incredibly dynamic and plastic, meaning they can change their function and behavior in response to the different stages of the reproductive cycle, pregnancy, and disease. For example, cells that form a tight barrier early in pregnancy can transform to help the cervix soften and open for delivery. One of the most important roles of the cervix is maintaining its structural integrity. This is largely thanks to a network of fibers, primarily collagen, which provides strength, and elastic fibers, which provide flexibility. Think of it as a finely woven basket that is both strong and pliable. During pregnancy, these fibers must be perfectly organized to keep the cervix closed. Research shows that if this organization is disrupted, if the weave of the basket loosens, the cervix can weaken and dilate prematurely, leading to preterm birth. Furthermore, the review highlights that the cervix has its own unique immune system to protect against ascending infections, which are another known cause of preterm labor. By understanding which cells are involved in this immune response and how they function, we may be able to prevent or better treat infections that put a pregnancy at risk. What does this mean for patients and doctors? The knowledge gained from single-cell analysis is paving the way for a new era of personalized medicine in obstetrics. The ultimate goal is to develop biomarkers, molecular signals that can be detected in a screening test, to predict a woman's individual risk for preterm birth or even cervical cancer. This would allow clinicians to move from a reactive approach, managing preterm labor once it starts, to a proactive one, identifying at-risk individuals early and offering preventative treatments. While this technology is still in its early stages, it offers immense hope for reducing the rates of preterm birth and improving the health of mothers and babies worldwide. Ma L. and Amp Nalasamy S. 2025. Cervical function in pregnancy and disease, new insights from single-cell analysis. American Journal of Obstetrics and Gynecology 232-4, 459-469.